Hello, we're glad you've joined us for this live webinar, ProQuantum Immunoassays Enable Low-Level Cytokine Detection. I'm Xavier Gutierrez of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Thermo Fisher Scientific. Thermo Fisher is a world leader in serving science, whose mission is to enable customers to make the world healthier, cleaner, and safer. For more information on our sponsor, please visit www.thermofisher.com. Now let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. And if you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Magnus Molin, PhD, a senior scientist at Thermo Fisher Scientific. Dr. Molin graduated from Uppsala University in Sweden with a master's degree in biology and a PhD in medical virology. He followed this with a postdoctoral fellowship at the University of California, San Francisco. Dr. Molin is now a senior scientist at Thermo Fisher Scientific, focusing on product innovation as a part of the Immunodiagnostics Division. For more information on our speaker, please click on the Speaker Biography tab at the top right of your screen. Dr. Molin will now begin his presentation.
Hello, my name is Magnus Molin, and I am a senior scientist working on developing new tests for respiratory disease such as asthma. Because we needed new ways to measure low levels of inflammatory proteins, we used the new ProQuantum high-sensitivity immunoassay in our research using clinical samples. And this is the work that I will be presenting today. So I would like to give first an introduction to respiratory diseases with a focus on asthma, and then describe the assay workflow for the ProQuantum immunoassay that we used. I will present some data using clinical samples, where in one case we looked at a cohort representing wheezing children. In another case, we looked at asthmatic children, and both of these cohorts included age-matched healthy control groups. Finally, I want to mention some results we obtained from analysis of nasal and fecal samples. Some background to respiratory diseases, such as asthma and COPD. So these are figures from the European Lung White Book based on statistics collected within the European Union. It has been estimated that asthma and respiratory diseases altogether cause 6 million hospital admissions and 600,000 deaths every year in the European Union. And total cost for this is around 380 billion, whereof 200 come from asthma and chronic obstructive pulmonary disease, COPD, which is a closely related disease. It has also been estimated that 1/3 of the population will develop asthma at some time. The problem with asthma is that it is a heterogeneous and complex disease with multiple clinical phenotypes. So currently there is no really good biomarker for asthma diagnosis, prognosis, or therapy response. As you can see in the right-hand side of the picture, showing the bronchial and healthy airways, asthma is characterized by airway inflammation and swelling. And you oftentimes get an increase in mucus production and typically a narrowing of the airways, which altogether makes breathing much more difficult. So there are several approaches to phenotyping of asthma patients. And there is evidence for genetic predisposition to asthma, so there may be reason to do genetic or gene expression analysis. It is also well established that asthma involves activation of the immune system, and the disease is characterized by the presence of immune cells in the airways. Histologic analysis of airway biopsies may be used to look at the extent of inflammation and airway remodeling. More recently, studies have shown that there is also a role for the airway microbiome in asthma development. From a clinical perspective, attention is typically focused on lung function, assessing, for example, bronchial hyperresponsiveness. And of course, important information is also can be collected by questionnaires to asthma patients. Focus of this talk will be on the inflammation part, highlighted in red. This schematic cross-section of the airways shows the major mechanism of asthma progression and severity. Asthma may be triggered by exposure to allergens or pollutants, or be caused by viral or bacterial infections. Depending on the trigger, different pathways will be activated, involving different cell types and cytokines. The boxes that you see in the picture indicate research areas that will benefit from further investigation, and as you can see, there is a lot to study. On your right-hand side of the slide, you see some approaches to identifying and measuring molecules involved in airway inflammation. Mass spectrometry is the approach that allows for analysis of the greatest number of analytes at the same time, and is typically used as the first screening method. Candidates from a mass spec analysis can then be verified using, for example, a bead-based assay, such as the ProQuantum Immunoassays. For single target validation, traditional methods like ELISA are typically used, but for sensitivity purposes, we have chosen to use the new ProQuantum High Sensitivity Immunoassay. ProQuantum assays were recently introduced a little more than a year ago, and the menu selection has rapidly increased. While there are many more kits for human cytokine, chemokine, or growth factor targets, the menu for mouse is starting to expand. Although I only work with human clinical samples, I can really appreciate the need for those with mouse models working with small volumes that do need a greater sensitivity. Targets included in the studies that I will talk about are indicated in bold in the table. Compared to other immunoassay platforms that my colleagues have used, I find that the main advantage with ProQuantum Immunoassays is the high sensitivity, which enables us to analyze many more samples using a minimal amount of sample, and altogether the protocol is much simpler and fairly quick. Therefore, in the current study, we wanted to address one problem that clinical researchers face, the volume limitation of working with biobanked clinical material. For research purposes, clinical samples are typically only available in small volumes, and this is especially true for samples from children. With conventional immunoassays such as ELISA, sample consumption is relatively high, and this sometimes limits the number of biomarkers that can be targeted. So the scope of this study was to try and select a panel of candidate targets supported by the literature as relevant for respiratory diseases on our clinical samples. And one intention was to see if ProQuantum Immunoassays would allow us to measure multiple protein markers using only small volumes so that we can make more use of precious samples. Importantly, we wanted to see also if sensitivity was sufficient to measure basal levels of low abundance biomarkers. And regarding the focus, serum samples in the context of respiratory disease was at the center. But we also looked at nasal samples representing respiratory allergy. And as I will talk about at the end of the presentation, in a separate project, we looked at fecal samples representing irritable and inflammatory bowel disease, two conditions that are typically assessed via fecal samples. And we used ProQuantum assays to see if those could also handle these more difficult and complex sample matrices. Blood plasma and serum are very complex mixtures of more than 10,000 different proteins ranging in concentration by roughly more than 10 orders of magnitude. It has been estimated that less than 10% of all human proteins have been measured in a quantitative manner, and those are typically the most abundant ones. So the challenge is really in detecting those low and ultra-low abundance proteins that are at sub-micromolar concentration levels. And just to give you an example, on the left-hand side, I've indicated the typical sensitivity of two diagnostics assays that we run on our ImmunoCAP platform. We use them for detection of proteins involved in allergy and asthma. And as you can see, the detection limit for ECP and tryptase is around 1 nanogram per milliliter. And just to compare, I put the reference as a reference the measuring range of one ProQuantum assay, the IL-6 assay, and its measuring range. What is interesting to us is how well the ProQuantum platform complements our current systems by allowing us to go down in concentration and measure low abundance proteins. This enables us to look for biomarkers, in this case, cytokines that are relevant for respiratory diseases. So some words about the experimental details of the results that I will present. So we use ProQuantum assays that are listed on your right-hand side, and we selected them based on their relevance for airway inflammation. And some, like IL-6 and IL-8, are considered markers of neutrophilic inflammation, while others, like IL-5, IL-13, and eotaxin, associate with eosinophilic inflammation, the two main types of inflammation behind asthma. Interferon gamma, on the other hand, is typically triggered by viral infections and could be a very nice complement to the other markers. As for the instrumentation, we use the QuantStudio qPCR instrument for the readout and the cloud-based ProQuantum software available at Thermo Fisher site at no cost. And I will go into further detail on why this software, the app, may be very handy and useful. Serum samples were in all experiments diluted tenfold. Some words about the clinical samples, the serum samples. We analyzed a total of 168 human serum samples. And these were from two independent cohorts, where one cohort represented wheezing children 0 to 6 years old, and the other cohort represented older children that all had the doctor's diagnosis of asthma. Both of these cohorts included age-matched healthy control groups. The wheezing cohort was analyzed using 6 ProQuantum assays, while the asthmatic cohort was analyzed for 3 targets with a known involvement in so-called type 2 asthma. So this is the basic workflow for the ProQuantum assays. The assay format is based on PCR amplification and use of real-time PCR instrument, which is why the first thing one needs to do is to program the qPCR instrument. We should say that any qPCR instrument should work for the assay, and the temperature profile is a universal one, so you can use the same program for all assays. The lab protocol is prepared using the ProQuantum app that I mentioned, and it is available at the Thermo Fisher website. So using it, you print the lab protocol, and then you follow the steps of the protocol, either as a digital copy or as a paper printout. Here I would recommend when preparing, setting up your assay that you do use electronic micropipettes, preferably multi-channel ones. So when the assay plate has been set up, you simply put it into the qPCR instrument as you would for any typical qPCR run, and you start the run. You can then conveniently follow the progress of the run from your cell phone or your PC, depending on what's most convenient. I think it's worth briefly walking you through the ProQuantum app, showing you how to design a customized protocol. From the Thermo Fisher webpage, one have access to Thermo Connect, which is the portal for all cloud-based apps, including the ProQuantum app. You open the ProQuantum app and start by clicking Create Experiment. Next, you enter the catalog number for the kit you're using, and this will autofill the entire page. You can then choose to also manually add the lot number for the kit you're using, and after clicking Next again, you enter the standard setup wizard that autofills standards based on the number of replicates and serial dilution interval that you defined. Clicking Next will take you to the plate layout, where you further define the plate layout by entering the number of samples and fold dilution, and the plate setup wizard will complete your assay plate. Next, you will then save the plate setup to the Thermo Fisher cloud. And as a result, in the last picture, number 6, you will have the entire instructions for carrying out the assay summarized in a 2-page document. And you will have the option to print the lab instructions to bring to your lab bench. It is most convenient to simply place the lab protocol printout on the lab bench and just follow the steps of the protocol when setting up the ProQuantum assay. So briefly here's how the assay workflow works. What you do is to thaw and prepare the assay reagents, dilute your samples, and prepare a serial dilution of the standard. This is done on a working or setup plate, which is simply an extra plate you use to organize reagents. Preparation of assay reagents and dilution of standard is a straightforward and simple process. And as for the preparation of samples, it's oftentimes equally simple, but maybe a little more complex if you do a spike and recovery experiment. So from this setup plate, you then transfer your assay reagents, standards, and samples to the assay plate. And the final step is to start your run in a qPCR instrument following the temperature profile mentioned at the end of the protocol. ProQuantum assays utilize a proximity ligation technology, where the assay combines the specificity of high affinity antibodies with the signal detection and amplification capabilities of real-time PCR. The assay is carried out in two steps. First, as you can see on your upper left-hand side, you perform an analyte binding step where you combine sample and detection antibodies and incubate for 1 hour at room temperature on your lab bench. During this time, the analyte-specific antibody pair will bind to your molecule of interest. You then add qPCR reagents and place the assay plate in your qPCR instrument, where you perform the second step on your upper right. This step comprises a ligation event and a qPCR readout. A few words about sample consumption. Typically, you dilute serum or plasma 10 times. Out of this, you use either 2 or 5 microliter per well. Assuming you use 2 microliter sample, one kit allows for analysis of 48 samples if you run your samples in triplicate. And this means that you will only use 0.6 microliter of your original sample in total. And this is what attracted us the most. If you would compare it to an ELISA, you would typically use 25 to 50 microliter rather than those 0.6 microliters. And from a personal perspective, I think once I got used to it, this protocol is very simple and quick. And I have had a colleague run it, and she agrees as well. And in our experience, we run typically using one instrument up to 3-4 plates per person a day. Another experience that we have is the importance of using multi-channel micropipettes, preferably with the stepper function. And the reason for using multi-channel pipettes is obviously because you handle many more samples in parallel. The stepper function makes setup faster and more accurate in our experience. So again, this is our strong recommendation. So a few words of what's happening inside the qPCR instrument. Following the initial analyte binding step, which is carried out on your lab bench, the subsequent ligation and amplification step is done in such an instrument, and this picture shows the typical temperature profile that you use. In the upper left part of the picture, you see the 2 analyte-specific antibodies as shown before, and those are linked to 2 different DNA oligos. And what happens when these antibodies bind to the same molecule is that the DNA oligos will come into close proximity. And if you also have a splint oligo complementary to both of these DNA oligos, a duplex structure will form, and you have the option of amplifying this following a ligation event by a DNA ligase. And the resulting molecule will be amplified using those primers shown on the right-hand side, including a TACMA probe that will give the fluorescent readout. So with the TACMA probes, the amplification is monitored in real time, as you can see this schematics example on your right-hand side. And the PCR cycle values when the signal rises above a threshold will reflect and be used as a determinant of the protein concentration, similar to how you use cycle threshold values for quantitation of DNA or RNA with conventional qPCR. Some words about the data analysis flow. So once your run is finished, you have the option of choosing at least the instrument we're using to automatically upload your result to connect on the cloud. And once there, you open your ProQuantum app, and using the ProQuantum app, you combine the plate setup file that I described previously with the result file generated now with your qPCR run. And then you have support for doing result analysis within the app. If you want to do a more advanced analysis, you have the option of working more with your exported files. You can export PDF, Excel, and graph files to your local computer. So coming to our actual observations. This slide shows the results from our ProQuantum analysis of the wheezing children cohort, where we looked at 7 different human targets. Graphs show groupwise comparisons done using GraphPad Prism. Each graph shows wheezing children compared to the healthy control group, and red lines indicate median values. Scale on the y-axis is logarithmic. Prior to ProQuantum analysis, we measured the levels of a protein called eosinophil-derived neurotoxin, or EDN, using a research assay we have on our Immunodiagnostics platform. And the reason for doing this is that EDN is a marker of eosinophils, and we have previously observed an increase in this protein in wheezing children as well as asthmatics. EDN values are represented in the graph in the upper left part of the slide. And we were curious to look at the extent of correlation between EDN and the ProQuantum cytokines. So as a result of our comparisons, we observed an increase in IL-5 and IL-13, agreeing with EDN levels and the role in type 2 airway inflammation. We did not see an increase in eotaxin, a chemokine that attracts eosinophils. Moreover, we observed an increase in IL-6 and IL-8. As mentioned previously, those are more markers of neutrophilic inflammation. And for interferon gamma, a marker of viral infections, the difference between groups was less clear, with the greatest spreading concentration for the wheezing group. We wanted to look more closely at this, so therefore we looked at correlation plots to see which of these targets correlate on a group level. So what we could see is that while there's no correlation at all between EDN and cytokines in healthy children, some of the targets seem to show a correlation and an increase correlating with EDN for the wheezing children group. And trying to dissect this, in the next slide, we look more closely at the ones here, looking more closely at the ones that do correlate. We could see that IL-6 and EDN for the wheezing group seem to correlate. As mentioned, IL-6 is considered a marker of systemic inflammation, and it's also known to be a risk factor for development of severe asthma. There also seems to be a correlation between IL-8 and EDN. And it's also interesting to note that both of these analytes are produced by airway smooth muscle cells. If we look at the correlation between the different ProQuantum targets plotted against each other, we find some interesting observations. Again, scales on the axis are in this slide logarithmic. So while both EDN and IL-5 showed an increase for the wheezing group, they did not clearly correlate. However, both seem to correlate to IL-6, as seen in the graph on the left-hand side of this slide, and combined with IL-6 EDN graph on the former slide that I showed you. And as for eotaxin, if you remember the former slide, there was no clear increase in eotaxin when comparing wheezing to healthy children. However, as you can see on the right-hand side, for the wheezing group, there seemed to be a correlation between eotaxin and IL-5. And we believe that in order to further investigate this, we will need to use some multivariate approaches to shed more light on the complex interactions and correlations between all these molecules involved. So moving forward to the results from our ProQuantum analysis of the asthmatic children cohort. This was a slightly older group of children and all had a doctor's diagnosis of asthma. Similar to our comparison between wheezing and healthy children, we did group-wise comparisons. And again, we used EDN, a research marker on our Immunodiagnostics platform, as a reference. And this is represented in the leftmost graph. For the asthmatics, we anticipated an increase in so-called type 2 inflammation marker. This is why we focused this analysis on IL-5, IL-13, and eotaxin. The role of IL-5 is known to be in eosinophil activation and proliferation, while eotaxin is a chemoattractant recruiting neutrophils to sites of inflammation. And asthmatics, especially children, typically have an increase in eosinophil numbers, both locally in the airways and in the bloodstream. So therefore, we were a little surprised to see that the levels of IL-5 and IL-13 on a group level were actually lower compared to the healthy control groups. However, I should say that the spread in IL-5 and IL-13 values was greater for asthmatics compared to healthy, making conclusions a little more difficult to draw. And there were a few individual cases where the cases were higher than the controls. For eotaxin, however, there was no clear difference between the groups. We speculate that the reason we did not see a clear increase in, for example, IL-5 could be due to the fact that these individuals are treated, these asthmatics are typically treated with inhaled corticosteroids. And this treatment is known to reduce the number of eosinophils. And of course, this could affect the outcome of the experiment. Looking at the correlation between EDN and ProQuantum targets, we find some intriguing observations. Again, the scales on the axis are logarithmic. There seemed to be a negative correlation between eotaxin and EDN, as you can see on the left, while there seemed to be a positive correlation between eotaxin and IL-5. This result is perhaps not intuitive from the previous group-wise comparison, and as mentioned previously, we believe we will have to use a multivariate approach to further resolve the complex interactions between molecules involved, and hopefully clarify the pathways involved in airway inflammation. I want to finish this talk by mentioning some results we obtained trying to analyze two other sample matrices other than blood. This slide shows measurement of ProQuantum targets in nasal mucosal lining fluid. The nasal fluid was collected before and after a grass allergen challenge in which the allergen, a grass extract, was sprayed into the nose of one individual at time 0. Nasal fluid collected at the indicated time points were then analyzed for these 5 analytes. Interestingly, we could see a dramatic increase in IL-5 and IL-13, as you can see in the upper part of the picture, following the grass allergen challenge. This would indicate an activation of the type 2 inflammation pathway. And while the results from the other cytokines were perhaps less clear, at least IL-6 also seemed to respond in a dramatic way following this allergen challenge. In my final data slide, I just want to mention that we also explored the possibility to look at an even more complex sample matrix, fecal samples. So that's why we analyzed human fecal samples for the presence of IL-1 beta and IL-8. Protein was measured or extracted before measurement using a dedicated stool extraction kit. And using this extraction procedure, it seems that both analytes were readily measurable. What we find interesting is that the results, although the number of individuals is perhaps low, the results indicate that these assays are potentially sensitive enough to measure basal levels and therefore potentially able to discriminate between these two different gastrointestinal conditions. Coming to my conclusions, most protein assays like ELISAs would require typically 25 to 50 microliter sample. With ProQuantum assays, we use less than 1 microliter, even if we did replicates. So to answer our own question, can we do more measurements or more analysis using these ProQuantum assays? The answer is yes. The way we did this was to try select the targets on 2 cohorts representing 2 airway diseases, wheezing and asthma. We also applied ProQuantum assays to allergy research, in which we looked at nasal samples following an allergen challenge. Finally, we applied 2 of the assays on gastrointestinal diseases and fecal samples. I should mention that we used a specific dedicated stool extraction kit for sample preparation. You can find more details about this on our webpage. Altogether, these analyses, of course, need to be repeated to verify these results and preferably on independent cohorts. But so far, we think the results are promising. More targets are being launched, and additional ProQuantum assays may add to the ability to discriminate, for example, asthmatics from healthy. With this, I want to thank you for your attention and leave room for questions.
Thank you, Dr. Molin, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Dr. Molin will answer as many questions as time permits. Okay, the first question is, did you need to handle your sample in any specific manner for ProQuantum?
Okay, so to answer this, the answer is that no, there is no real difference between sample handling for these ProQuantum assays compared to, for example, ELISA. One recommendation, though, is that if you see any particulates or any aggregates, you should give your samples centrifugation. You could. So we actually tried this, but the way we handle our samples was just use them as is. We did not have any pronounced problems with particulates, but a recommendation if you want to be precautious is to do a centrifugation step prior to analysis.
And are you able to detect other cytokines than the ones in the example here, in particular R-spondin and WANT?
Sorry, can you repeat the question?
Are you able to detect other cytokines than the ones in the example here, in particular R-spondin and WNT?
Okay. So basically the ones we worked with so far are the ones presented here. So we didn't look at R-spondin or WNT.
All right. And what's the dynamic range of the assay?
Dynamic range is if you can remember, for some assays, the measure, I think I gave one example for IL-6, right? The measuring range was from 0.06 picogram per milliliter up to, I think it was 1,000. So that would be like, I don't know, 5 log.
And can this assay be multiplexed, multiple analytes detected in each well, or is it singleplex only based on the antibody pair per analyte per well?
I think the answer is that it is a singleplex assay, but I mean, technically you can envision, of course, in the future a multiplex version, but I don't know if this is on the table for those developing for future analysis. But as a user, I would say it's a singleplex assay.
Okay, and what is the typical analytical CV for these assays?
So we, in our experience, you get excellent CVs for standard curves, typically below 5%, I would say. Sometimes you see a little higher variation with biological samples, but I think that is true for any assay. And it could be due to what we talked about before, particulates, aggregates occurring in your samples. So I would suggest that you work with using repeated, and you make sure, I mean, the way we worked with our samples was to make sure that pipetting was really accurate, that we handled samples the same way at all times, and also that you try to avoid freeze thawing. I mean, if you do freeze thawing, some of the analytes will be affected by that. Cytokines are typically, according to the literature, they are sensitive to freeze thawing cycles. So my answer would be to keep those recommendations, and we'll have nice comparable CVs to other assays like ELISAs.
And are custom antibody pairs conjugated with the oligos available for analytes not listed on your website? And can such antibody conjugates be created on request?
So while I'm not part of the ProQuantum assay development team, I know that there is a plan, or it's in the process of being available, a service activity. So I think you can, at least in the future, you will be able to order a custom assays.
And are there any wash steps before PCR setup?
No, so the advantage with the ProQuantum assays is that different to ELISA, you don't have any wash steps. So there's this first analyze binding step, and then you have this combined ligation amplification step within your qPCR instrument.
And can the platform be used also for gastrointestinal inflammatory?
Yeah. So so the data, the preliminary data that I showed in my final slide, suggests yes to that. I mean, the answer is yes to that question. So that's specifically what we looked at: two different gastrointestinal inflammatory conditions.
And do you need special qPCR instrument, or is Roche Light Cycle 96 will be suitable?
So we didn't try any Roche instruments, and I never worked with the light cycler, so I can't tell. But we used the Quant Studio 5 as mentioned. So, but according to the information I have, the app and the platform should be open to any standard qPCR instrument.
And is there a concern of antibodies or protein analytes being denatured in the denaturing and annealing steps of PCR cycles, or is the contribution of antibodies only in the initial ligation step, and does it not interfere afterwards?
So while I didn't do the development work, my guess is that antibodies will not interfere as after DNA oligos have been ligated and the duplex formation occurs. So the answer is antibodies only play a role in the first step.
Okay, well, that looks like that's all the time we have for questions right now. I would like to once again thank Dr. Molin for his presentation. We would also like to thank our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through August 2019, and LabRoots will alert you via email when it's available for replay. We encourage you to share the email with your colleagues who may have missed today's live event. Until next time, goodbye.